We want to present for you our investigation of the bathing water quality in Norway, location Roggesvei. This is our location, Norway. It's located approximately 9 kilometers outside of Bergen, towards Flesland. We zoom in and we see Norway. This is our location, referred to as Roggesvei. Norway is recognized by its stratification layers, and it's a brackish water system. But first, I want to take you back to 1969, the changing point in Norway's history. For many years, the municipality put in a lot of sewers in 2 main sites into Norway. This was done after a theory to keep the sewage on the bottom layers, like in the basins, because of the stratification would keep it down. This was in theory. So, the winter of 1969, the ice was as normal, covering the whole surface of Norway, except for a little part. And here, the lipids got the sewage to rise up to the surface under the ice, killing almost everything on its way, and made a little nice surprise for come spring. When the ice melted, there was dead organisms everywhere, and it was really smelling. So the inhabitants around Norway thought this was over. But actually, it was the opposite. The paradox was that this actually helped Norway get re-oxygenized in the deeper layers. And from the quality has actually been improving. So the second paradox is that to maintain this quality, we need to keep these 2 inputs from the main inputs from the municipality to keep the water column oxygenized. Back to our site in Roggesvei. We were actually 2 groups on the same site the same day. So we had to divide the area into 2 parts, divided by the red line, you see here. This is our site in a better map. We chose 4 sites, where site 1 and 3 was an inshore site, and site 2 and 4 was an offshore site. So we could compare offshore water to inshore water. At site 1, there was a closed down sewage outlet, and site 3 had an open sewage outlet. Site 1 and 3 is comparable to last year's results, since 2 and 4 is new. To get there, we had to use a boat to sample the offshore sites. So Thursday morning, I woke up and I drove out to Gjellestad to drive the boat into Norway. So I cruised into this view over here, eating my breakfast, and it was very nice. Started the day. And our aim of this investigation was to assess the overall bathing water quality in the area, and to assess the improvement in bathing water quality compared to 2011. To sample these 2 types of different sites, we needed different sampling equipment. So to sample the pH on both onshore and offshore sites, we used the pH meter. For the temperature and salinity on both sites, we used the conductivity meter. And for the biodiversity in onshore sites, we used kick sampling. And for the biodiversity on the boat, we used a van der Veen grab. For the TCB on the onshore sites, we used a rod and sterile bottle, and we did the same for the phosphorus. And on the offshore sites, on the boat, we used a Rotner bottle to do the water sample for the TCB and the phosphorus. Here you can see the Rotner bottle and the pH meter. And also the van der Veen grab and how we stored our samples. Now I would like to call upon Gustav to present our results and discussion.
Thank you, Jennifer. So now I will be discussing about the result and discussion. First of all, I will discuss about pH, temperature, and salinity. our results of pH and temperature did not show any significance because they were fluctuating. But however, our salinity results showed that salinity increased with increase in depth. This is due to this points out that there is stratification at this site. Now moving on to the biodiversity, the results of our samples of different sites are shown in the pie charts. We classified them according to the phylum. During the sampling process, we also caught some animals at site 3. For comparing with the last year's results was difficult because they had only qualitative data. So what we did is that we just wanted to make sure that we are able to compare the suitability of habitat at our 4 sites. For this, we used 2 indexes, Simpson's index and Sandow-Wiener index. And the results of those indexes showed that Site 3 and Site 4 are good habitat compared to Site 1 and Site 2. And Site 2 is, however, a good, not so good habitat. However, Site 1 was found to be a poor habitat. Now to continue on result analysis, I would like to call BIO.
Then we're going to look at the phosphorus results. These are the phosphorus results for the different sites. We can start at the top at Site 1. We see that the difference from last year is pretty high. And this is like Jeanette mentioned, there was a pipe there that closed in between our sampling last year and this year. And then it's Site 3 that we can compare to last year's. We see there is no significant difference, or actually they're pretty similar. Looking at the offshore sites, 2 and 4, we see that the phosphorus increased with the depth. And this points back to what Utsav said, that there's a stratification. So the phosphorus that comes into the water, it sinks down and it's kind of trapped under, because it's stratified, so it's trapped under the lasagna plates. No, the salt. You have the salt. Then we're moving to the TCB. Here we have a color scheme for the columns. Green is excellent, yellow is good, and red is poor. And the black one is some we made up, because it's the contaminated ones. We had some samples that were contaminated. And looking at the results from all the sites, taking site 1 and 3 first, we see that there has been a decrease in the TCB values. Yeah, I forgot something. The colors, or the classification is based on EU's directive from 2006. And then back to Site 1 and 3. We can see that there's a decrease in the values, even though the decrease at Site 1 is a bit lower than the decrease in, or it's not as good in Site 1 as Site 3. Looking at the 2 new sites, the offshore sites 2 and 4, we had 6 samples at each site, and this is the mean, and it's excellent. It's actually pretty low. It's really low. So, summing it up for the phosphorus and the TCB count, we would say that Site 1 is good. Site 2, 3, and 4 are excellent. But we have to keep in mind that our contaminations from the bottom samples had some sediments in them. That's why they were contaminated. So that means that there's a lot of TCB in the bottom. So if you're going to swim there, you shouldn't dive, because you can stir up the sediments, and yeah, there's a lot of TCB there. So yeah, just don't touch, only swim.
So our further recommendations for the area is maybe we have prepared information brochure. That will be an example to mail out or hand out to the residents around Roggesvei. It's important to remind them to think when they fertilize their garden, so they don't put additional nutrients into the surrounding water. And it's also very important to empty the septic tanks regularly, so they don't overflow when it rains. The solution is clear. Now I would like to call upon Daniel to give us a global perspective.
Okay. How we can benefit from Roggesvei experience in global perspective. In Sudan, I came from Red Sea State in Sudan. Pollution from many resources is measured according to reef conditions. Also, many studies confirm that coral reef cells very good, considered good with 30% or to 50%. Water quality for basin in Sudan not priority to our government. It is kind of luxury, but to me as a biology student, I know it is very important to make water investigation, and especially for our lovely kids, to give them clean beaches, and also for people who are interested in diving in warm water of Red Sea. And
Now in global perspective in terms of Nepal, so these are the pictures showing the water quality in rural areas on the left side and in the urban areas on the right side. The right picture is the river close to my house, and the quality of water was not always like this. So when I was a child, I used to go swimming and fishing there, but now I cannot go there. So I would like to say is that our government or in our country, the priority is given to drinking water. But for me, I think I would love to have priorities given to bathing water quality as well. So these are our references, Ritu. Thank you.
Thanks very much. Really good.